Alright, welcome back to our lecture on hematopoietic stem cells. Well, that was the last lecture that we had, so today we're going to be talking about a different highly regenerative tissue, and that is the liver. And the liver has been very interesting and a bit elusive in terms of stem cell research because it's really regenerative, right? You can remove 60% of someone's liver and it'll grow back, which has led researchers to go, wow, that's probably stem cells, because that makes sense in terms of the way we think about stem cells. If you've got something that's regenerating, it feels like it's stem cells. But that action in particular actually isn't a stem cell function, and we'll go through that. So today we're going to go through a little bit of liver anatomy, then we're going to go through, what we do with the stem cells, and as usual, we'll look at the therapies. So let's get into it. So you've got a thing called like I said, the regrowth of that liver is just a lot of proliferation from adult cells. It's not stem cells creating new cells, but we'll go into how the stem cells actually come about. All right, so let's get some liver anatomy. So you have a liver lobule, which I'm not going to bother to draw because it's not that important. But what we are going to draw is a bile duct. Right, so a bile duct. Right, and the bile duct, and there are two main kinds of liver cells, okay? Two main kinds of liver cells. There are hepatocytes and cholangiocytes. So the hepatocytes live down here, and they perform a lot of your liver function. And cholangiocytes live here around the edge of your bile duct. And out here is your hepatocytes. And these are cholangiocytes. These are your cholangiocytes. And cholangiocytes make your bile. Cholangiocytes make bile. They live around there, they make bile. They live around the bile duct, makes sense. And then your hepatocytes live over here and in the rest of the organ that's not directly around the bile duct. And they perform other functions of the liver. But you may have noticed that I've cunningly not drawn anything in this area here. Now why could that be, Ryan? Why could that be? Well, this is a very special area called the canal of herring. Canal of herring. Write it down. Canal of herring. Important. Remember. Important. Okay? Canal of herring is geographically in between the hepatocytes and the cholangiocytes. And in terms of its shape and its behavior, it's kind of in between the two as well. So this is our hypothesis for where the stem cells in the liver reside. Now, we don't call them liver stem cells, we call them liver progenitor cells because naming conventions. I don't have a good reason for why that is. I think they're stem cells. But like I said before, there's been a lot of controversy over whether or not they are indeed stem cells. And so the fact that we call them progenitor cells is likely to be an artifact of all that debating. Okay, so, like I said before, once we take away a big chunk of the liver, right, and it regrows, that function, that regrowth, is not a function of stem cells. It's a function of grown-up cells, adult cells, proliferating. And so we need to figure out how do we get these stem cells to come out and do things? How do we get them out? How do we find them? How do we refine them? How do we differentiate them? Okay, and so an important thing to know is that when we take a hepatectomy, so you take, 2/3 of that liver from your donor, with a sharp scalpel and a professional surgeon, there's not a lot of signals going to the cells saying, there's an injury here, we need to do something about the injury by regrowing with stem cells, okay? There's not a lot of signaling. And so in order to get that signaling, and in turn to get the stem cells to come out and play, we need to injure the liver in some way. Okay? And so that's done in a variety of ways. And we can talk about, you know, having hepatitis or drinking really bad and getting some genuine liver damage. But in order to get the stem cells to come out, and this is the only time that stem cells come out, is when you have serious damage to the liver. So we took note of that fact. Stem cells only come out when we have legitimate damage to the liver. And so now we need to damage the liver in order to get the stem cells to come out and be stem celling. Cool? So I'll just draw it very quickly. Right, no, that's crap. So that's the same diagram we drew before. And normally you just got your cells hanging out around the outside. But if you damage the liver, right, you get the proliferation of these liver progenitor cells coming out, damaged. They come out around here and they're ready to repair things. So let's talk about that in terms of an experiment. So, like I mentioned, they come out when they're damaged, and we noticed that when we damaged the liver, there was an increase in Wnt signaling. Wnt signaling. And we went, wow, we know how Wnt signaling works, and we can even detect cells that are doing a lot of Wnt signaling, and we can pull them out using LGR5+. LGR5. And so what LGR5 allows us to do, and there's another pathway, and but in short, we use LGR5 to allow us to pull out cells that are exhibiting Wnt signaling. So we take a sample of cells, and the experiment applies CCl4 to them, which is a chemical which damages the cells, and it causes some Wnt signaling to occur, which we can detect. And when that Wnt signaling occurs, we know that it's only occurring in the stem cells, and so we pull out those cells which are eliciting the Wnt signaling, which are doing the Wnt signaling, using LGR5. Makes sense? Damage the cells, cells get damaged, and they do the Wnt signaling, and when they do the Wnt signaling, they have LGR5, we can pull them out using LGR5. Cool? Cool. Yep. Now that we've pulled those cells out, we put them into a plate. Put them in a plate. And they have demonstrated biclonal potential. And I think I'm off the page. Oh, not quite. Right, and that's cool because it means that the cells we've pulled out are indeed stem cells. Okay, now that's real cool because that tells us how do we actually get the stem cells out of the liver. Now we can talk a little bit about therapy with the liver stem cells. So what do we want to do with liver therapy? There's a comment in my notes that says 60 to 80% of liver cancers are hepatocellular carcinoma, if you care. So we want to be able to model liver disease, right? Model liver disease. We want to be able to correct genetic defects. We want to be able to avoid the need for donors. That's a big one. Are you in focus? I hope so. Avoid the need for donors and finally, we want to have a whole lot of cells for toxicity testing. Fucking be in focus. Okay? So those are the things that we want to be able to do with our liver stem cell therapy. Now the thing about liver stem cells when we culture them, or liver cells in general when we culture them, we want to make organoids, okay? 3D organoids. And liver cells really don't like being in 2D culture. It cramps their style. It's not their vibe. Not their vibe. But if we take them in 3D culture, they actually grow really well. And it works for humans and mice, right? So we do the thing that we did before. We damage the mouse liver. We get out the stem cells using our LGR5. We get out and we grow them, and you can grow them in 3D culture, and you get a little organoid, and then you can put that organoid back in your mouse, and you can get something close to curing liver disease, which is pretty cool. And so, yep, you just need to make sure you start either with those stem cells or with a fragment of canal of herring. Canal of herring. Yep. Cyst-like structures. I'm going to pause you again. Okay, and the last thing I'm going to say is about human organoids, and you just need to make sure when you're doing this with human organoids is that the human stem cells are more like cholangiocytes than they are hepatocytes. More like cholangiocytes than they are hepatocytes. More like bile duct cells, bile duct cholangiocytes. Than hepatocytes. So we need to gently coerce them towards being a mixture of cholangiocytes and hepatocytes. Otherwise, if we just grow the human stem cells and we take a little sample of the bile duct and get some duct structure, if you just take your little sample of duct structure and you try and grow it, what you'll end up with is cholangiocytes. You won't get hepatocytes. So we need to gently coerce them towards being hepatocytes, but not to the extent that we would get only hepatocytes. And that is the liver. Thanks, guys.